I might actually be the only person still working who's been through all three generations of drug discovery. After 50 years of it, I think I know even less than I did 50 years ago.
Dave Brown helped invent one of the most famous drugs in history, and now he's reinventing how we discover new ones. He was part of the team behind Viagra at Pfizer, led R&D at Glaxo and Roche, and today is the co-founder of Helix, a Cambridge-based company using AI to accelerate treatments for rare diseases. Join my colleague Saranga as he talks to Dave about 50 years of drug discovery, what it took to keep his discovery alive inside a giant organization, and why he believes technology can remove human errors in the way we do science itself. This is What's Next, conversations with the founders and CEOs building the future.
So Dave, thank you for coming in. It's great to have you here. I was just reflecting that we've spent 7 years now working together. I first invested in Helix back in 2018. One of the things I've really enjoyed about the 7 years that we've worked together is that I know that you're a very broad and deep thinker, not just about your own field of drug discovery, but also much beyond that. But before we kick off into all of that, I thought maybe we could start with just a bit about your background. Could you just talk us through, where you started in your career and what you did in all the years before you started Helix?
Okay, so my first degree was in chemistry, and I specialized in organic chemistry, physicochemical properties of molecules, which then leads into drug design, of course, and quantum physics. I did quantum physics alongside my chemistry. I've always been a bit crazy that way. Then for PhD, I was dead lucky because during my first degree, we had a visiting lecturer who turned out to be the head of chemistry for what's now AstraZeneca. And, within 5 minutes of him starting talking, I knew what I was going to do with my life. As soon as he started talking about drugs and designing them, wow, that's what I want to do. Yeah. So that really motivated me to get the top first that I needed for the PhD. But during the PhD, I taught myself to design drugs. It wasn't the main topic to start with, but I sort of segued into that as soon as I could.
Why were you drawn to the problem of designing drugs? Was it the intellectual challenge? Was it the impact you can have on patients, or a bit of both? Or
all those things. It was really reducing human suffering. It was that sort of feeling. That's what I want to do with my life. But I spent my childhood building things basically, and then I began to realize. Chemistry was a bit boring to me, quite honestly. It's too easy, but designing molecules to interact with the molecules of life, if you like, enzymes and receptors, that looked like a decent challenge.
Yeah, there's something really interesting about having complete control over the thing you're designing, but then having to interact with something you have no control over, right? It's a sort of unknown meets the known sort of problem.
Yeah, and after 50 years of it, I think I know even less than I did 50 years ago. It's a lot more complicated than we ever realized for lots of reasons.
So maybe we should talk a bit more about that. I want to hear more about your career soon, but before that, just looking at the world of drug discovery, and you've been involved in that industry for a number of decades, how has it developed? What are the sort of big themes or trends that you've seen in your career?
Yeah, I might actually be the only person still working who's been through all three generations of drug discovery. It was just called drug discovery when I started it. It was ICI Pharmaceuticals then. It then sort of got renamed as Zeneca and then AstraZeneca at Alderley Park in Cheshire, a wonderful place to work. It was just called drug discovery. We now call it phenotypic drug discovery. This was the first generation, which basically meant you make a molecule in the chemistry lab, give it to a biologist, injects it into an animal, and you see what it does, but you've got no idea how it's working because we didn't understand human biochemistry in those days. That's now called phenotypic. And then we started to get equipment in 1985, 86, 87 that allowed us to fractionate chemicals themselves. In fact, the Viagra project was a pivotal point. It was when we first got that equipment, we then got the drug. We would have failed if that equipment hadn't come along. But yeah, we got big advances in human biochemistry that began to isolate biochemical targets. So the industry by the 1990s has switched into what's now called target-based drug discovery, where you know the enzyme or receptor you want to go for. and you design a molecule for it. Hasn't been wonderfully successful, I have to say, for a lot of reasons. But now we are moving into the third generation. It's the age of big data, multi-omics, as we call it, is what we're doing in Helix. And I think we've got to merge all three, actually. Otherwise, you just fragment those different methods. And that's the kind of vision I have for the future.
And that's a really good point, because when we first invested in Helix, we spent time with probably 50 or 100 different sort of third generation drug discovery companies. And we felt like many of them really were so convinced of this third omics, multi-omics approach that they completely discounted the value of what came before.
Yes, exactly.
Whereas, as you know, when we talked to you, we thought there was a really great balance between excitement about the new tools, but also some understanding that the old tools had relevance.
Yeah, this is a really important point. I've always been a bit of a techie. Believe it or not, I've been involved in what's now called artificial intelligence since my undergrad days. I was programming in Fortran, self-taught, and during my PhD, I was doing what's now got the fancy title of multi-parametric linear regression analysis, which is just algebra, basically. That's the first generation of AI, we call it now.
Yeah.
And there's no doubt that each technology takes 15 to 25 years to come to a point of being useful. And yet people latch onto it thinking, we've got this, it's going to pay back in 5 years. In fact, AI has taken even longer than that. Yeah, 1960s were the first wave. We're now in the fourth wave by my account, and now it's beginning to work. As we can see in the world in general, that's taken 50 years. Yeah. So technology takes a lot longer to pay back than most people realize.
So going back to target-based drug discovery, one of the iconic, if not the iconic, case study in that era globally was Viagra, and you were the lead in that project. Tell us a bit about how you ended up working on Viagra and what that journey was like.
Yeah, so I had 11 very good years at what's now AstraZeneca. Loved the place. Pfizer had advertised a good position at their sort of growing research labs in Sandwich in Kent. And I didn't apply for it because I thought it was too senior for me. But then I got approached by a headhunter a year later saying they hadn't filled the position. Went for interview. I was seriously impressed with the quality of the people who interviewed me. And so I took the job, even though Pfizer was totally unknown in this country at the time. It was a fraction the size of Alderley Park, of course. Alderley Park had the cream of the scientists in the country. Everybody wanted to work in drug discovery, wanted to work there. That's how good it was. But anyway, I took the risk. The very first thing I worked on was the Viagra project.
Okay.
It didn't start that way, of course. There was a research proposal being written to lower blood pressure by a particular mechanism. And we started on that. Went nowhere for 3 years. I was designing molecules, using just physical models and computational chemistry, and really making no progress. The project was very badly resourced. Only 2 or 3 people in chemistry and 2 or 3 in biology. It was the least resourced project in the whole place.
Interesting. Okay.
All the way through. It stayed that way, actually.
Yeah.
The management didn't value it. And then there was a paradigm shift, basically. As I mentioned earlier, the development of the new technology that allowed us to start to fractionate biochemicals from human cells came along in 87, 88. There was a professor at Glasgow University, Miles Housley, who had the first equipment in the country. Okay. We approached him and funded a PhD student who kind of half solved the problem. But then we took the technology into our own biochemistry labs, and within a year we had Viagra. It wasn't called Viagra then, but what happened is we went from phenotypic, in other words, just injecting things into rats as we used then, to actually knowing what the target was we were aiming for. And that's transformational for anybody designing a drug molecule.
Right. So it's, to sort of think about it in a high level for those of us who are not computational chemists, it's that special equipment allowed you to really improve
isolate the enzyme. Okay. There were 3 different enzymes we were interested in.
Right.
And we didn't know which was the right one, and we didn't have a way of testing against them either.
Right.
Then we discovered the particular lead molecules we were getting very good activity in the rats hit one of those enzymes,
Okay.
phosphodiesterase 5, PD5 it's called.
And then you knew the enzyme that mattered.
Yeah, and then we were able to optimize the molecule very quickly after that.
Right.
Now, this is really important in terms of history of drug discovery because it was that pivotal point between first generation and second generation, phenotypic and target-based. And Viagra was sort of half phenotypic, and then we did some of the very first target-based, and that allowed us to get the molecule.
And so one of the things that I've never quite understood is, as you said, when the Viagra project started, when you joined, it was aimed at blood pressure. And then obviously...
Angina was next.
Right. And so how did it end up being, the impotence product?
Yeah, 8 years of pain.
Okay, yeah.
God, it was awful. I mean, they were trying to kill it many times.
Okay.
I was getting crucified every review meeting, more or less. And
Why? Because the progress wasn't there?
Well, yeah, very slow progress. We didn't know the target to start with. There were other projects that they believed in much more that had all the funding. The technology wasn't ready.
Yeah.
We started looking for a molecule with lower blood pressure. When we had a molecule we could put into rats, it looked more like a nitrate that's used for angina, the profile. So we switched into angina as the clinical target, selected a molecule that has the code name UK92480.
Okay.
And they started calling me Mr. UK 9480 because I was kind of defending this molecule to the death for years, kind of thing. That's what it took. Then, and I designed that molecule, and I was inventor on the patent of it as well. And then we selected that to go into clinical trials for angina, and nobody in the clinical group wanted to lead the clinical trials. They didn't believe in it. They thought it was a waste of time. So they let a chemist lead the clinical team.
Okay. Yeah.
Never happened before. Purely because nobody wanted to do it, which was kind of transformational in my life. So I was very fortunate in that sense. We nearly lost the drug twice in the first 6 months. We sent it to an external laboratory for safety testing, and they sent the data saying back it killed all the animals. We knew that wasn't correct
true, yeah.
because, the effects were so bland and so safe. We had the opposite concern.
Right.
So we just knew it was wrong, but they wouldn't admit they'd sent us the wrong data. Okay. And after about 6 months of arguing with them, they admitted they'd sent us another company's data, not ours.
Okay.
Yeah. We got through that. And then when you scale up synthesis, to make enough to put into humans, there's a risk because the chemists change the way you make it to try and simplify it and make it scalable. And what happened was they suddenly hit a crystalline form that was totally insoluble. And they couldn't go back to the original one. Totally insoluble means it's not going to dissolve in the stomach. It's not going to get absorbed.
Not going to get absorbed, right, yeah.
So it took about another 4 months to solve that. And we changed the salt of the molecule. And then it was back on track again. But we lost a lot of time in that first year. Nearly lost it twice. It was all very bland during the initial dosing to humans, no problems. We did a lot of studies. I was kind of shocked when I went back through all the clinical folders. I think we did 20, 25 studies, because again, we were struggling to show any effect at all. Anyway, it went into angina patients. And I remember sitting in my office one day thinking, well, they're just setting me up for failure here. They hadn't given me enough money to dose enough patients to show it would work because they just didn't believe in it, basically. It was just totally set up for failure. And then once a quarter, I had to present to the head of Pfizer Europe and the seniors. And there was one meeting. He said, David, you've wasted money for 8 years. If you don't get some good data now, we're going to close it next meeting in 3 months' time. Close.
Okay.
And it was literally a few days after that.
Yeah.
The clinical research associate running a study in Wales came in my office and said, the men are reporting erections all night.
Okay.
Now, these weren't angina patients. These were volunteers because we weren't sure that we'd got the dose right. So we were doing sort of dose response studies. Yeah. I must say she blushed bright red, and I'll never forget that. She was so embarrassed by all this.
Because it was unexpected as well, right? So,
And so I got the the two seniors on my team together. It was literally, just a corridor just outside my office, and we talked it through and instantly understood why it was working. There'd been some science published 9 months earlier that made sense to us.
Okay.
So we understood why it could work. But more than that, probably the most important thing was that about 3 years earlier, believe it or not, a colleague and I had proposed to Pfizer, seniors in the US this was, that we work in male impotence. And we had spent ages working with the commercial people in New York City on doing a commercial analysis. We knew there was a huge unmet need and massive, multi-billion market. Now, they were talking about 4 billion sales, which would be, this was 1980s.
Yeah.
Imagine what that is now. 4 billion is, I don't know, what, 10 or 20 billion sales.
Yeah.
So that was the
like
market
15
size.
Yeah.
And they turned it down. We presented to the head of research and he said no. But the important thing is we'd done the market analysis. We knew there was a huge need there. So when the young lady came to my office and I talked to my two colleagues, I immediately went to the head of clinical and said, look, there's this huge market. We've done the analysis.
Yeah.
We've got a drug that works in patients. We think it will. Now, remember, these were volunteers.
Yeah.
They weren't impotent males.
Yeah.
So we had to do the study. And we understand the science now. So, give me some money to do a clinical trial. And he said no. And it was one of those kind of pivotal moments in my life. I stood up, went and closed the door of his office, it was open, and said, I'm not leaving until you give me the money. An hour later, he gave me the money. And he took a big risk himself because his budget was under pressure. He'd been in that meeting that he knew the senior management didn't want the drug. Anyway, he gave me the money. We did 2 studies in Bristol, Southmead Hospital, Bristol. I think it was the only impotence clinic in the country. There was only one in the US at that time as well, in California. First study was 11 or 12 men. The second was about the same. You just couldn't miss it. I mean, the effect was just nearly everyone responded.
Right.
Yeah. Yeah, we got funded to do more studies. At that point, because we'd proven it worked, they took it away from me, which was one of the most shocking days of my life, and put a clinician in charge at last, which hurt terribly. But it was the right thing to do because, you've got to be very expert to sort of deal with the regulatory authorities and scale up these studies. So a young clinician called Ian Osterloh took over. Yeah, and it was a very rare drug in that, we were getting 70, 80, 90% response rate. You never get that with any drug. So it wasn't proving efficacy that was the problem. We had enough data with 300, 400 patients. It was the safety. So the time was really spent on getting into about 5,000 people just to prove it was safe.
So that's a really interesting thing in itself, right? I mean, obviously, we work together because I backed a startup. When I remember meeting you the first time, I was intrigued by your background, but I thought of you as someone who'd worked in large companies for most of his career, which you have. But actually, the dynamic sounds incredibly similar to being in a startup. You've got a small team. You believe passionately that you've figured something out, and you keep having to go back and ask for more money. And sometimes the people with the checkbooks don't hear and don't listen.
But to be fair, most people in big companies don't do that.
Yeah.
And it was only after Helix was my second or third startup.
Right.
That I actually began to understand myself. I'm really a small company entrepreneur.
Yeah.
And I think the reason it succeeded in Pfizer was I was behaving like a, very determined small company entrepreneur.
Yeah.
But, most people in big companies will just bow to their management and not risk their career.
Yeah.
Yeah.
So you had this incredible journey. We all know how it ends. With Viagra being, a pivotal product for Pfizer and generating, billions and billions of dollars of not just revenue but profit as well, and in fact continues to be a key product for that company, one of the largest pharma companies in the world. By that point, you've already gone to Glaxo and you're working there and you head up their research as well. You then have a long and extremely successful career in the pharma industry. And at some point, you get to that point of age where you decide to retire, and this is the bit that sort of continues to intrigue me. You've been incredibly successful. You've got all these fantastic notches in your career bedpost, why at that point didn't you just, buy the big house in the country, buy the sports car, worry about the grandchildren, that kind of thing, play a bit of tennis, I don't know what it is. But, instead you went on and did startups. Why on earth how did that happen?
Age 54,
Yeah.
my wife sat me down for the first time and said, "Oh, yeah, what happened after Glaxo is I went to Roche. It was Roche I was head of their research globally in Switzerland. Very good company, Roche is. Then I ran a startup for several years, Cellzone, that we sold to Glaxo. But I had to raise a lot of money, after the dot-com crash in year 2000, 2001. And I was traveling the whole time. And my wife just sat me down and said, look, I calculated you were away 36 weeks last year.
Okay.
Yeah. We don't need the money. Why are you doing this?
Yeah.
So she made me retire. So age 54, I retired, which seemed good to start with. And I did a year of meditation because I've always done a lot of meditation. And it was a wonderful year, I have to say. Two things happened. One was after 6 months, my wife said, David, I'm not used to you being home the whole time.
So she regretted her move. Okay, yeah.
So I thought, okay, I've got permission to work again.
Yeah.
The second thing that happened was I really actually did miss the people, the work, and the creativity. That's what I just, I get a buzz from working with smart, nice, creative people and generating ideas, and I was just missing that. I needed that to go back to that. So I volunteered to help Bill Gates actually with his third world diseases. This was Bill and Melinda Gates Foundation, but it was before Bill actually moved in. He was still at Microsoft at that time.
Okay.
It was small in those days.
Yeah.
We got $43 million from them working on diarrheal diseases and babies because there were 2 million baby deaths from diarrheal diseases. Did that for a while, for 4 or 5 years. Then started chairing Baberham Research Campus, which you'll know is...
It's outside Cambridge, yeah.
It was nothing then, it was just, there was one new building basically, but it's now completely full of new buildings, an incubator site, biotech site. I chaired that for many years. And as part of that, spun out a company called Crescendo Biologics, antibody company, and chaired it for 6 years. Very tough 6 years, but it's doing fine now. Tim came and found me, 2013.
So you met Tim Gilliams,
yeah, that's
your
right.
co-founder at Helix in 2013.
Yeah, what happened there is I'd been helping another Cambridge entrepreneur start a crowdfunding company, Syndicate Room. Jonathan Milner and I were the first investors in it. And because I'd helped him, Tim had an office next to him in the Cambridge Enterprise incubator.
Right.
So he said to Tim, go and talk to Dave, he'll help you get the company going. So we spent a year mostly in the alcoves of the Judge Business School trying to figure what we're going to do, meeting and talking. And at that point, I switched to Helix and have been doing it ever since.
And you... But what's interesting about Helix is you didn't just chair it or sort of advise or invest. You got involved. I mean, you were...
Well, I was the only scientist in the company.
Yeah,
for many
so you
years.
were working full-time as one of the co-founders.
And unpaid for many years.
And unpaid, yeah. So what was it about, I guess, the combination of Tim, your co-founder, the idea, why did you do it?
If you look back on your life, there are some pivotal moments, aren't there? And there was one in the chemistry department at Cambridge. Andreas Bender, who was also a co-founder with us, it turned, as a result of that meeting, we've been talking to him about various genomics technologies, and he and a couple of students were writing some code, which was the very first foundational method for Helix in the end. But we went in, listened to him and his student present it, and a light bulb went on. I thought, wow, this is the way to do it in the future. So then I agreed to start the company with Tim, and we took that very crude code. We had to completely rewrite it. It took us 6 months. and got going on that basis. So that meeting really inspired me to think this is now third generation drug discovery. We can do things in a way in which we take the human bias out. Because what happens in drug discovery, I describe phenotypic, target-based.
Yep.
What happens with target-based is that a biologist has a view of which biochemical target should treat which disease if you modulate it.
Yeah.
It's wrong 95% of the time.
Right.
And you can always come up with preclinical evidence that kind of supports your theory if you try hard enough, and that's what happens. With this technology, the omics technology, I could see that you could do things in a very different order. You could take the human bias out. You could let the data tell you which drug should work, and then work back to the mechanism. So you've actually reversed the order of things, and that's exactly the way it worked out. It took several years. I mean, our first staff were recruited in 2015. Then 2017, it was only computer programmers plus Tim and me. So I was the only sort of scientist drug discoverer. So I did the projects myself. We did several projects in 2017 and 2018, and every one of them was stunningly successful with this technology. Our success rate in predicting which molecule should work when we took them into the lab studies was 50% or something, which is just impossible. Really.
Right.
He almost,
staggering.
it was a shock to me, quite honestly. I didn't expect it to be that good. So at that point, you came along, and we scaled the company, and made a huge difference.
Yeah. And so let's talk a bit more about that then. So, as you say, with targeted drug discovery, it still required sort of human intuition, the first step, right? You look at all these options and you say, I think that could be an interesting, place to bind to, or, part of the biological system that I want to change. And as you say, 95% of the time people are wrong. Let's talk a bit about multi-omic, data-driven drug discovery. How does it, as simply as we can describe it, how does it work? Fundamentally, how does the philosophy work? Obviously, the technical details are evolving all the time, but...
Well, what I'm going to describe is the way we do it.
Yep.
Other people are doing it in lots of different ways.
Sure.
Because this is still very exploratory.
Yeah.
As I said, technologies take 15 to 25 years to mature, and we're probably only 10 years in.
Okay.
So I think by next year, we could have the first evidence that this technology works.
Yeah.
I think we will literally be the first, actually, because most of the competitors have fallen by the wayside now.
Yeah. So again, how does it work at Helix? What does the approach do?
At the moment, we've got, I'm losing track, 12 or 13 different algorithms. We started with only one or two. And they all work in very different ways. And I haven't got enough time in the next 3 hours to describe all 13.
That's fine. Yeah.
I'd probably get it wrong anyway.
Yeah.
But for instance, the algorithm that we took out of the chemistry department and improved, that predicted a current cancer drug. The way it works is you look at the difference in expression of DNA, which is the RNA, the difference between disease state and normal state. So the DNA will overexpress some things and underexpress some things.
Right.
And that's giving you a picture of what the difference is between disease and non-disease.
Yeah.
Okay? Then we look for a molecule that does that, squashes down or raises up those genes, the expression of those genes, the function of the genes, back to the baseline.
Right.
Simple concept.
Yeah.
This concept came out of this genius Italian guy, Francesco Aure. I don't know if you've ever met him. No. He helped us initially. He was working at Hinxton Genome Campus, published a paper in Drug Discovery Today 2013, which is one of my absolute favorite papers ever, describing this method.
The method of actually mining the data to find the match, is that what you mean?
Yeah, that's right. So what you do is you gather cells. So for instance, the cancer that we've been working on. You gather cancer cells from patients. Because it's a monogenic disease, a single gene defect causes the cancer. You can put those into mice and replicate it in mice. And you gather the cells from the mice. And you can do it with single cells as well, but actually having that in vivo data is even a lot more powerful.
Right.
You then analyze what's happening in those cells, what's overexpressed and underexpressed. You then generate a profile which you match to drug molecules. You do exactly the same with drug molecules. You look at what a drug does overexpressing or underexpressing the RNA in a cell.
Yeah.
And you want the drug to be the opposite profile, mirror image
Yeah.
of the disease state.
Yeah.
It's a simpler concept as that.
Which is exactly what these statistical machine learning type approaches work really brilliantly at, right? Having a really large universe of these and then figuring out which one is the one that's the match.
I mean, I actually, because I'm working on it again, I was in the office last week and I said to the team, well, actually, if we worked on this for all our careers, we could do it manually with spreadsheets, but it would take that long.
Yeah.
But with the computers, we can do it in days.
Yeah.
So it's just number crunching on a massive scale, basically. So we did that, several projects in 2017, 18, many of them succeeded, and it was like, wow, this works. At that point, I had a lot of confidence in it.
Yeah. It's great. So let's, if we can, talk about some other things. So, you've sort of hinted at this before, but you're very interested, I think you've had this amazing career where you have, whether by design or by luck, probably a bit of both, been at that pivotal moment where drug discovery has changed 2 times, right? From phenotypic to target-based, and then from target-based to multi-omic, and sort of data-driven, if you can call it that. This has led, I think, partly to the fact that you're very interested in how we think and how we solve problems and how we think about things like logic and so on. I mean, tell me a bit more about that and your interest in that area and what you think the sort of lessons are for all of us who are involved in anything to do with trying to solve, what comes next in the world.
I think people are all very different, and very few people actually have the freedom in their mind to do this kind of thing. This phrase human bias, I think is really important. I'm actually writing a book on scientific method at the moment. And the undercurrent all the way through it is how human bias is damaging scientific method in many, many ways. I think I've only used that term once in the whole book, but the current is there. What happens is, we have actually this came to me when I was doing some intensive meditation a few years ago, and then I thought, I'll go on with writing this because I could see there were lots of problems. There's a kind of triangle between the concepts we have in our mind, the way we perceive the world, concepts in our mind, the thoughts we generate, and the way we perceive the world that feeds back and reinforces our already established concepts. It's kind of obvious when you think of it, and people assume that scientists can escape from that. No.
And somehow be objective.
That's right.
Yeah.
So the whole point of scientific method is to put in place procedures
to guard
against it. But they've been degraded terribly in the last 50 years. 30 years later, things are even worse, I would say. As I mentioned, I was presenting at the Genome Campus on Tuesday. Professor Ross Clark stands up and says exactly the same thing. He started, all through his half-hour presentation, he kept saying, science is being degraded because this is happening. And it's exactly the same things as I've written in my book. So there are people aware of this. And it's pretty difficult to escape it. But what I want to do, one of the things I want to do, I'm going to try and get somebody in the university to work on it, is we can write a machine learning algorithm now. If I've produced a diagram of what scientific method is, this kind of cyclical process, what the weaknesses are at each step, what the defenses are at each step. We can program that into a machine learning algorithm and feed in grant applications, research proposals, papers for publication, and it can tell us where we're making mistakes. This is pretty easy to do.
Yeah,
So it's
yeah.
one of the things I want to do in the next year, work with people at Cambridge University on this.
So this is what's interesting to me, because your point is that the scientific method, if you take it in its sort of purest form, is a very smart construct which was designed to remove the biases that we all know humans bring to any of these endeavors. Machine learning, on the one hand, is huge promise because it's a machine, so you can tell the machine not to bring bias and to sort of follow the rules explicitly and so on. But the downside, what we're seeing a lot with AI right now, for example, is that machine intelligence is trained on a lot of historic human behavior and data. And so guess what? It replicates the bias that can...
And it's even worse than that. Again, this is a whole chapter in my book. Have you come across paper mills?
No.
No, most people haven't.
Yeah.
And until I started studying it in the last couple of years, I hadn't either. There are so many fake journals. I'm getting emails every day from fake journals asking me to submit. There was a Nature, in one of our top science journals, published in 2000 and I can't remember the year, 2 or 3 years ago, saying 10,000 papers had been proven to be false. That's just the tip of the iceberg.
Right.
It's tens of thousands.
Yeah.
All this is out there, and LLMs are being trained on it.
Yeah.
All this false science. So it's a very serious issue.
So how do we fix it?
Well, as I said, I think we've got to produce an algorithm that can detect false science and can detect whether science hasn't been done in the appropriate way. Part of the problem is statistics. This p-value of less than 0.05.
Yeah.
Most scientists don't understand statistics. They just outsource it to somebody else. And you can fiddle the statistics so easily. Yes, if you're interested in this, I can send you the Nature paper, or I can send you the chapter of my book.
Yeah.
It's really fascinating what happened. The frequentist approach was never designed for the way it's being used at the moment. The whole thing was just corrupted terribly. I want to get this book published, and I want to keep it brief because I think every PhD student has to read it to understand, to avoid the obvious mistakes that are being made.
If we can just completely switch gears, this is a different topic. It has struck me that you've had a career that spanned multiple countries and continents. You've worked here in the UK, obviously, but also in Switzerland, also in the US, for a variety of different cultures. You've spent the last few years in Cambridge and really embedded in that particular sort of innovation ecosystem, and very much on the life sciences side, which is something that Cambridge does really well. There's a big debate, it's a never-ending debate about how we can do a better job in this country, in the UK, but also in Europe, in creating an innovation economy that can drive the kind of growth and economic benefits that we see in places like the US. What's your take on that? We do so much fantastic science here. Why do we seem to stumble sometimes at translating that into economic value?
In fact, I've got a whole presentation on this, which you've never seen.
Yeah.
2008, 2010,
Yeah.
I did a 2-year contract with the EU, working with PricewaterhouseCoopers in Luxembourg.
Right.
They won the contract and signed me up as a consultant to them. And what had happened is that the EU in 2004 to 7 had been approached by the East European countries that had left the Soviet Union and joined the EU, saying, "Hey, give us some money and we'll build biotech hubs." And the people in Brussels quite rightly said, "Well, it probably takes more than money."
Yeah.
So they had us analyze what they call the CSFs, critical success factors, for biotech clusters around the world. We analyzed 16, ranging from marine in Japan to San Diego and whatever, Silicon Valley in the US. I wrote the report on the Cambridge cluster. So we did agriculture, healthcare, and marine. I actually gave the final presentation in Brussels. And in fact, I gave the presentation last October to another conference, and it's still absolutely correct. We analyzed 6 different factors. One obviously was finance, another was culture, another was sort of industrial base skills. Do people want to live there? The tax regime, all these things. And it was pretty clear. It was a bit like a three-legged stool, except it was a six-legged stool, if you see what I mean. If just one of those was missing or was weak, it stood in the way. So that analysis is still worth looking at.
So Dave, you've now been in the drug discovery world for just over 50 years. Look forward for the next 50 years. What's the biggest breakthrough going to be in those 50 years?
Well, AI is, there's no doubt. 10 years ago, I'd have said it was hype. But we've had 50 years of development of AI, another 50 years, and it's just getting to the point where actually it's impressive. Another 50 years, wow. We can't even conceive today of what it will be able to do. So there's no doubt about that. Some people say it's scary. I think it's a bit like the Industrial Revolution in the 1800s. Instead of Industrial Revolution, mechanizing, manual labor, this is now mechanizing, if you like, computerizing intellectual endeavors. I think we're going to see the same massive improvement in productivity. Back then, 70 or 80% of people worked on the land.
Yep.
It's now 2%. Yeah, I mean, what's that, a 30 or 40-fold improvement in productivity?
Yep.
I think we're going to see the same on the intellectual side. I just wish I was 30 years old because I'm really excited about it. And I think it's just at the point of you can do something magnificent with it. Yeah, so I think it's going to be AI. In terms of healthcare, what do we need? We need healthy aging more than anything, and I think there will be breakthroughs there. There's research ongoing, but obviously it's a very difficult and lengthy process, to try things and see what works. We particularly need breakthroughs in Alzheimer's and dementia because as people live longer, they say everybody will get, if they live to 120, Alzheimer's. That is probably only a slight exaggeration, unfortunately. So, if the average lifespan is going to be 100 years, that's what they're saying about babies born today, on average will live to 100 now. There's going to be an awful lot of dementia. So I think we need breakthroughs in that more than anything.
Great. Thank you so much, Dave. It's been absolute pleasure, obviously, working with you for the last 7 years, but then also sitting down today and talking about everything, not just Helix, but your incredible career and the way that the sort of drug discovery market has developed and morphed over that period. Thank you very much for your time.
Yeah, thank you.
Thanks for listening to What's Next. We hope you enjoyed the episode, and if you want to learn more about the frontier of deep technology in Europe today, please like and subscribe. And if you're interested in learning more about working in deep tech, look at the Balderton website to learn more about us and our portfolio companies.